Okay, today we are going to look at RNA interference. RNA interference is like a mechanism where the gene expression at the state of translation is being inhibited. Like for example, here look at it. Remember in the central dogma of molecular biology, we have DNA, and the DNA is converted to mRNA, and then the mRNA will be translocated or will be transported from the nucleus to the cytoplasm. So the transcription is usually taking place in the nucleus, while the translation is usually taking place in the cytoplasm. And remember, the ribosomes is a protein manufacturing like industry, is a protein manufacturing industry. That is where the proteins is usually synthesized. So the mRNA in the nucleus synthesized must be transported from the nucleus to the cytoplasm where the protein synthesis occur. So now, if we now look at this, we'll now see that the mRNA will be converted to proteins through a process called translation. But here, when we have an RNA interference, the DNA, look at it here, the DNA is converted to mRNA, and then the mRNA is now being inhibited or is now being deactivated so that the protein synthesis will not take place. So that is why in a simple term, when we said RNA interference is like a types of the RNA that usually block the protein synthesis. So it inhibits the synthesis of a proteins by stopping the translation. So that is the meaning of the RNA interference. So in a simple term, the RNA interference is used to degrade or digest the mRNA produced from the DNA in the nucleus. So therefore now, whenever you have an RNA interference, it can easily block the translation, that it inhibits translation. That is the importance of RNA interference. Okay, and then now, the next thing now that we are going to look at, why do we need this RNA interference? Why do we need it? So we need RNA interference because of two major reasons. Number one, defense mechanisms, and number two, genome-wide regulation. So in the case of the defense mechanisms, remember that we have different types of RNA. So we have, different types of viruses. We have RNA virus and we have DNA viruses. So like HIV virus is an RNA virus, coronavirus is an RNA virus. So using RNA interference, you can block or you can protect the cells against the infection by the virus using this RNA interference. And then second, we have what you call insertional element. Apart from the genome itself, or in the genome itself, there are what we call insertional elements, or we call them transposable elements. When we say transposable element, it means that they are mobile genetics element. They are part of the DNA that have the ability to transfer themselves from one point to another. So using this RNA interference, you can protect the genome from the insertion of some genetics element in the genome that are not important to the body. So that is in the case of the defense mechanisms. So it means that in a simple term, you can use RNA interference to block or to inhibit or to protect the cells against viral infection by degrading or digesting its mRNA. And then second, we have genome-wide regulation. So in the case of genome-wide regulation, we can use RNA interference to investigate or to regulate the development and the genome. So you see, we have some genes that are important in terms of human development, like growth hormones or we have some proteins that we don't even know their functions. So therefore, you can use this RNA interference to be able to regulate or to understand the functions of a gene. Like now, for example, let's say you have your DNA here. So when you have your DNA, which is the double-stranded, and you have, let's say, a particular gene here, which is gene 1, and you don't know the functions of this gene. So if you can now be able to understand the sequence or the sequence of the nucleotide of these genes, then you can now be able to understand the kinds of the messenger RNA that this gene will be produced. So if you have that basic knowledge, then you can now design an RNA interference that is complementary to this mRNA, which will now be used to block the translation of this mRNA into proteins. And from there, you can be able to monitor the functions of the proteins that is produced by these genes. Okay, and then the next thing now that we are going to look at is the different types of RNA. So we have two types of RNA. We have short interference RNA, and then we also have micro RNA. Also have short interference RNA, and we have micro RNA. So these are the two types of RNA interference. So I think we are going to stop here and we'll continue in the next video.